the night a virus became family. It starts small. A bite, a scratch, a shared fruit. An ancient primate catches a retrovirus, the kind that doesn't just hijack a cell, it rewrites it. Most infections end in a dead end. The body clears the virus or the host dies, and the code goes with them. But not this time. The virus carries simple instructions. Its RNA genome enters a cell and using reverse transcriptase becomes DNA. Integrase threads that DNA into the host's chromosomes. Long terminal repeats, LTRs, stamp the start and end like bookends. In the middle sit the classic viral parts. GAG for the capsid shell, POL for enzymes including that reverse transcriptase and integrase, and ENVER for the protein that fuses membranes. It's a tidy kit for breaking and entering. Here's the 1 in a million break. The infection lands in a germ cell. Not a skin cell, not a liver cell, an egg or a sperm. That's the only doorway to forever. When that germ cell becomes a zygote, the integrated provirus hitches a ride into every new cell as the embryo divides. Suddenly, a one-night stand is a lineage. Within a mating season, that family carries a quiet passenger. Some insertions vanish by chance, others ride a wave of reproduction. If that retroviral scar doesn't kill, it drifts. If it helps, by changing when a nearby gene turns on, or by blocking entry to cousin viruses, it might even spread faster. Over generations, neutral mutations hit the GAG, POL, and breeding frames. Frame shifts, stop codons. The once infectious provirus decays into a fossil. But the LTRs, those regulatory bookends, can still talk to the host machinery. The genome keeps receipts. At each insertion site, you can see target site duplications, little footprints flanking the provirus. Some families of these elements prefer certain chromatin states, slipping into open, actively transcribed regions. Others pepper heterochromatin, like confetti in the silent parts of the genome. Either way, the insertion creates a new regulatory possibility. A promoter where none existed, an enhancer with a fresh transcription factor binding site, or a decoy that soaks up repressors. Meanwhile, there's a counterattack brewing. Host cells learn to spot viral hallmarks and clamp down. KRAB zinc finger proteins evolve to recognize specific sequences in these newcomers. They recruit TRIM28, also called CAP1, which calls in histone methyltransferases to lay down H3K9 Me3, heavy repressive marks. DNA methylation spreads like wet cement, hardening the silence. In germ cells and early embryos, small RNA pathways, pRNAs, guide machinery to matching sequences and warn, shut this down. ApoBec enzymes mutate cytidines into uracils on single-stranded DNA during reverse transcription, leaving pockmarks in any latecomers. The firewall rises. But firewalls with doors are useful. In the right tissue, an LTR can be a well-placed access point. Some LTRs carry binding sites for STAT1, the switch that flips when interferon floods a cell during an infection. One viral remnant family, MER41, seeded interferon-responsive enhancers near genes like AIM2, APOL1, IFI6, and SETIM1. What began as a threat became part of the alarm system. The host didn't just defend, it repurposed. Picture a troop of primates after that first season. An infant tugs its mother's fur. Inside its cells, the retroviral scar sits in the same chromosome region the mother carries, the same region the father lacks. In a few generations, by drift and chance, the insertion becomes common. If it's harmless, it may hang around as background noise. If it subtly boosts resistance to a circulating virus by interfering with its receptor, or if it reshapes expression of a nearby gene in a way that improves survival, it can fix in the population. Everybody has it. Now, it's not an infection, it's a locus in the species. Time blurs the line between self and invader. Recombination between the LTRs can pop out the interior viral genes, leaving a single solo LTR behind like a signpost. The two LTRs at the ends also slowly diverge from each other, ticking away like a clock that lets geneticists estimate the age of the insertion. Younger families, like HERV-K, still show relatively fresh sequences, and in some cases, residual activity. Older ones, like HERV-H, are nearly all fossils, but still lend regulatory phrases to the genome's grammar. There's a moment when this isn't just molecular, it's personal. The ancestor with the fever mates. A zygote forms. One cell becomes 2, then 4, then a blastocyst. The provirus copies with every division, cemented into the origin story of a person who will never know it's there. That scar, in one lineage, will one day regulate an immune gene during a winter outbreak. In another, a descendant's brain will open a set of LTR-driven enhancers at just the right developmental hour to sculpt a cortex subtly different from a cousin species. A threat became an option. Of course, not every passenger is a gift. Some insertions slice into the middle of important genes and get purged by selection. Others sit close enough to matter, but far enough to survive, waiting on the epigenetic leash. The host and the remnants learn a tense coexistence. Most sequences locked down, a minority kept poised ready for regulated use. This was the night a virus became family. Not by destiny, by mechanics, chance, and selection. What nobody knew in that forest was how far the consequences would reach. Because one of the greatest human upgrades would come from the most viral of tricks, fusing cells. And that story starts where every one of us started, at the placenta. Viral tools, human upgrades, exaptation in action. To understand the upgrade, remember the kit that virus left behind. LTR bookends with promoter power, and a cargo hold that used to make infection possible. Once the replicating parts broke, what remained was a pile of switches and sequences. Evolution doesn't waste a good switch. Here's the stakes check. If a viral remnant helps survival even a little, shaving a day off an immune response, nudging a developmental gene at the right hour, it's more likely to stick around. If it's harmful, layers of silencing pile on until it's effectively mute. Most are quiet fossils, but a fraction got hired. Start with the alarm system. During infections, interferon floods a cell and flips STAT1 and other transcription factors. Some ERV LTRs already carry binding sites for those factors, because the original virus needed to turn on fast when a cell's machinery was active. The host stole that bias. One ERV family, MER41, landed near immune genes and now acts like a row of fire alarms. When interferon spikes, MER41 enhancers help light up genes such as AIM2, APOL1, IFI6, and SECTUM1. What used to be a viral on-ramp became part of a human early warning network. Meanwhile, the brain found its own uses. Development is all about timing and place. Turn this gene on in this cell type for a narrow window, then shut it down and hand off to the next program. LTRs are natural candidates for alternative start sites and enhancers. In the developing cortex and hippocampus, clusters of ERV-derived switches open in waves, recruiting transcription factors that weren't even a concept when the virus first jumped in. The result is subtle. A tweak in neuron subtype ratios, a change in dendrite branching thresholds, differences that add up across many loci to tune learning, attention, and memory. It's not one dramatic lever, it's 1,000 sliders. Mechanistically, the adoption is hands-on. A dormant LTR sits in heterochromatin, silenced by H3K9, Me3, and DNA methylation. In a specific tissue, pioneer factors bind a motif within that LTR, recruit coactivators, and repel repressors. Nucleosomes move. The LTR becomes an enhancer looped to a nearby promoter through cohesin and CTCF, changing 3D chromatin architecture. Or the LTR's core promoter is repurposed as an alternative transcription start site, producing a longer or shorter first exon that alters translation efficiency or protein localization. Sometimes the LTR launches a long, non-coding RNA that scaffolds regulators to a neighborhood of genes, modulating an entire program. Get this, the immune system didn't just adopt the alarms, it borrowed viral tricks for blocking. Some ERV and remnants, though crippled, kept receptor-binding fragments. Expressed in certain tissues, they can act as decoys that occupy viral entry receptors, lowering susceptibility to related pathogens. It's a simple move. Jam the lock with a broken key. Across mammals, independent captures of NVA-like sequences keep showing up, hinting at a strong selective payoff. The payoff isn't only protection. Tissues under heavy regulatory demand, placenta, brain, immune cells, early embryos, show enriched ERV-derived regulatory activity. It's like evolution sprinkled accelerators. where rapid, context-dependent tuning matters. Humans and chimps share many ERV fossils, but the same family can be wired differently in each species. Different LTRs opened, different factors recruited, different loops formed. Over millions of years, that rewiring contributes to lineage-specific traits. You can see it in stem cells, where certain HERV-H elements mark active pluripotency networks in humans, more than in other primates, shaping how early embryos set up developmental competence. There's another layer: responsiveness. Because these sequences evolved to be sensitive to cellular states, stress, inflammation, hormones, they serve as built-in context sensors. Under interferon, a subset of LTRs wake to boost antiviral genes. Under neuronal activity, activity-dependent factors bind neuron-specific LTRs to fine-tune synaptic genes. Under hypoxia, a different subset may engage. The host didn't invent thousands of new switches from scratch. It sifted through the viral junkyard for parts already wired to respond. But there's always a but. Reusing hacker tools means living with hacker risk. The same LTR that boosts an immune gene in a macrophage could, if misexpressed in a neuron or a germ cell, create noise or toxicity. So the system evolved layers of breaks. KRAB-ZFPs that recognize particular LTR subfamilies, TRIM28 to enforce repression until the right factors arrive. PRNAs in germ cells to keep the heritable line clean. It's a negotiated truce where the host sets strict curfews and occasional work permits. And here's where it gets dramatic. Among all the exapted tools, one set stands out for changing mammalian life. Captured M genes that fuse cells. Viruses use MV to merge with host membranes. Mammals repurpose that fusogenic power to build a new tissue, the syncytia trophoblast of the placenta. In humans, two of these MV-derived genes took center stage with different roles and timing. We call them Syncytin 1 and Syncytin 2. Those names sound technical, but their effect is not. Without them, placental fusion fails, nutrient and gas exchange stalls, and live birth as we know it collapses. A viral battering ram became the architect of early life. We'll zoom into that fusion engine next. The placenta's secret: syncytins and live birth upgrades. Before there was a heartbeat, there was a merger. A tiny ball of cells reached the uterine wall and needed a way to plug in, share oxygen, share nutrients, and avoid getting mistaken for an invader. That plug isn't a simple connection. It's a living interface called the syncytia trophoblast, a continuous, multinucleated sheet that fuses from many cells into one barrier. And the engine that does the fusing came from a virus. Viruses use invric proteins to fuse their membranes with host cells, pry open a route in, and dump their genetic cargo. Mammalian placentas hijack that exact talent. In humans, two invade-derived genes stepped into essential roles with different timing and duties. Syncytin 1 from the ERV-W1 family and Syncytin 2 from ERV-FRD1. They aren't backups. They're co-leads in a choreography where a single misstep can collapse a pregnancy. Here's how it plays out. Trophoblast cells at the outer layer of the embryo differentiate into cytotrophoblasts that line up against the maternal tissue. Syncytin 1 is expressed on their surfaces. When its receptor aligns, like two halves of a zipper, the protein snaps into an active shape, brings membranes within kissing distance, and drives them to merge. Two cells become one, then another adds on, and another. The sheet grows into a syncytium, the fused skin of the placenta, studded with microvilli that increase surface area for exchange. A viral battering ram became a construction crane. Syncytin 2 runs a parallel job with quieter but critical effects. It also carries fusogenic power, but its expression pattern and regulatory control differ, and its protein includes an immunosuppressive domain, an ancient viral trick turned guardian. At the maternal-fetal interface, that domain tones down local immune activation, helping the mother's immune cells stand down in a place that, frankly, looks like a graft. Together, the two NV-derived proteins balance force and tolerance. Build the barrier, then keep the peace. The stakes couldn't be higher. If fusion fails or the immune truce cracks, conditions like preeclampsia, fetal growth restriction, or early loss can follow. Researchers have found altered levels of Syncytin-1 or Syncytin-2 transcripts and aberrant methylation around their LTR promoters in complicated pregnancies. It's a reminder that the genome's locks and keys aren't philosophical, they're medical. Now widen the lens. This wasn't a one-off miracle unique to primates. Across mammals, evolution kept stumbling onto the same hack. Rodents captured one NV gene for fusion, then another later. Ruminants like cows did it independently, bats did it, carnivores did it, different viruses, different times, same solution. Grab INV, domesticate it, and build a placenta that fuses. That convergence screams utility. When nature finds a working part in the junkyard, it keeps pulling the same part. This convergence hides a second lesson. There's more than one way to tune the same organ. Lineages that domesticated different NV genes show differences in placental structure and depth of invasion. Shallowly invasive placentas, like in some ungulates, solve nutrient transfer with surface area and vascular architecture. Deeply invasive primate placentas lean on intense remodeling of maternal arteries. The shared language is fusion. The dialects are species-specific. Of course, viral tools bring viral liabilities. The same fusogenic power that helps build tissue can get misapplied. In several cancers, melanoma, breast, germ cell tumors, HERV-derived NV proteins, including HERV-K and members of the HERV-W family, show up out of context. Cell fusion in tumors can create hybrid cells with new traits. More motile, more drug-resistant, more aggressive. Some M proteins can also act as ligands for innate immune receptors, like TLR4, kicking off inflammatory cascades that reshape the tumor microenvironment. What built the nursery can also help an invader break walls. So why doesn't the placenta fuse everything in sight? Locks, and lots of them. The LTRs flanking Syncytin-1 and Syncytin-2 carry promoter sequences that respond to placenta-specific transcription factors. And those same regions are kept repressed in most other tissues by DNA methylation and repressive histone marks. KRAB-ZFPs patrol for the wrong context. TRIM28 enforces silence unless the right factors arrive. Even within the placenta, expression is spatially patterned. Cytotrophoblasts express, syncytiotrophoblasts maintain, so fusion proceeds where and when it's safe. There's a microstory hidden in every pregnancy test. A few days after conception, as HTG rises, the embryo sends molecular messages to preserve the uterine lining. Meanwhile, on the cell surface, Syncytin-1 molecules are quietly zipping membranes together, turning hundreds of individual trophoblasts into a single working organ that will filter a mother's blood for months. No fanfare, no blueprint written by mammals from scratch. Just an old viral skill repurposed perfectly. One detail would matter later. These invade-derived proteins still look viral enough to the immune system to be recognized when they appear in the wrong place. That's part of why they can become red flags in tumors, and part of how medicine is starting to use them. But before we turn the immune system loose, keep one idea in mind. If a virus helped build the interface that kept you alive before birth, what else did viral code help wire after? The next clues aren't in the womb, they're in the brain. The firewall, how cells muffle the viral choir. We've been celebrating the upgrades, but upgrades come with guardrails. 8% viral code is powerful hardware. If it misfires in the wrong tissue, at the wrong age, with the wrong trigger, it can jam gene programs, fuse cells that should stay separate, or trip immune alarms. So cells built a layered firewall, fast, redundant, and paranoid. Start at the DNA. The simplest lock is a chemical tag, methyl groups glued onto cytosines, especially at CPG islands inside LTR promoters. Methylation doesn't change the letters, but it blocks transcription factors and recruits proteins that keep chromatin tight. It's the universal do not enter tape, renewed every cell division. Layered on top are histone marks. The key repressive badge here is H3K9, Me3, a trimethyl flag planted on histone H3 that tells packing proteins to squeeze that region into heterochromatin. Closed chromatin means the polymerase can't get in. The viral promoter can't shout. Who lays the flags? A specialized police force. KRAB zinc finger proteins, KRAB-ZFPs, are an enormous family of DNA-binding sentinels that evolved in part to recognize specific transposons and ERV sequences. Each KRAB-ZFP is a custom key that fits a handful of sequences. When it binds, it recruits TRIM28, also called COP1, which acts like a hub. TRIM28 pulls in SETB1, the histone methyltransferase that writes H3K9Me3, and it scaffolds other repressors and chromatin remodelers. In early embryos and stem cells, TRIM28 sits like a blanket over young retroelements. Knock it down in mice, and endogenous retroviruses lurch awake innate immunity sparks, and microglia in the brain flip into an activated state. That's not a theory, that's what labs see when TRIM28 is deleted during development. ERV transcripts rise, ERV-derived proteins appear, and inflammatory pathways light up. Germ cells get a second firewall, small RNA patrols. The PEER-ENA pathway, built around Pee-wee proteins, uses tiny sequence guides to find transcripts from transposable elements and ERVs and slice them up. It's ruthless because any slip here becomes heritable. Eparinase also direct DNA methylation machinery back to matching loci, reinforcing the lock at the source. In somatic tissues, other small RNA systems and RNA interference catch strays, while RNase H chews on RNA-DNA hybrids that form during errant reverse transcription. There's also a sabotage unit that scars incoming invaders. Apobec enzymes, cytidine deaminases, rip Cs into us on single-stranded viral DNA during reverse transcription, causing hypermutation. Those edits cripple latecomer retroviruses and leave pockmarks even on ancient remnants that attempt to reactivate. If you read an ERV locus and see an unusual pattern of mutations, chances are Apobec took a swing at it long ago. None of these locks acts alone. The genius is in layering. Most ERV copies sit under multiple forms of repression: DNA methylation, H3K9, Me3, KRAB-ZFP TRIM28 complexes, small RNAs. So if one mark erodes, the others hold. But not all marks are equal everywhere. Early embryos loosen global methylation, then reseal it with help from TRIM28 and SETB1. Placental cells deliberately open select LTRs. Immune cells hold certain interferon-responsive LTRs, poised, repressed, but close to ready, so a pathogen signal can safely crack the door. The catch is that locks weather. Aging erodes methylation fidelity, the pattern phrase. Chronic stress and inflammatory cytokines can tug at chromatin. Interferons, the same molecules that help fight viruses, can also flip on ERV-linked enhancers like MER41 to boost antiviral genes. That's by design in macrophages, less so in neurons. In diseases where global methylation is reduced, like many cancers, swaths of ERV loci unsilence together. Double-stranded RNAs from antisense sense ERV transcription pile up. Sensors like MDA5, TLR3, and the She-gas sting pathway smell virus and trigger interferon programs. In tumors, that can be a feature. We'll use it to make cancers visible. In healthy brain tissue, it can become a fuse for neuroinflammation. And the brain firewall is delicate. In mouse models, when TRIM28 is deleted in neurons or during development, ERV activation correlates with microglial activation and expression of ERV-derived proteins in adult brains. The innate immune response doesn't care that these sequences have local citizenship. To sensors, viral nucleic acids are viral nucleic acids. That tension, citizen by birth, suspect by pattern, defines the entire relationship. Context matters. In the placenta, Syncytin 1 and Syncytin 2 LTRs are demethylated and opened by trophoblast transcription factors. In nearly every other tissue, those same promoters are locked down. KRAB-ZFP repertoires differ between species and even individuals, shaping which ERV families stay quiet and which can be coaxed for regulation. That diversity can be protective, more keys for more doors, but it also means some people's firewalls might be leakier at specific loci, changing risk profiles for autoimmunity, infertility, or cancer. Think of it as a choir most of us never hear. Thousands of voices, trained to stay silent, with a few soloists licensed to sing in carefully chosen rooms. The conductor is epigenetics. The ushers are KRAB-ZFPs and TRIM28. The soundproofing is methylation and H3K9, Me3, and the bouncers are PEERNAs and Apobec. Most nights, the hall is quiet and the show runs on time. But a power surge, aging, infection, chemotherapy, stress, can pop a circuit. A handful of mics switch on. Sometimes that noise becomes a life-saving alarm. Sometimes it becomes harmful feedback. We'll see both. First, the harm, when ghosts wake and push biology out of tune. Then, the gambit, using that very noise to flag tumors for destruction
and disease.